I'd like to welcome everybody to our side event entitled Using the Power of Endocrine Science to Evaluate the Endocrine Disrupting Effects of POP Substances. And I would like to thank the Secretariat especially for making this possible. We really appreciate it. And I would also like to acknowledge the three organizations that are sponsoring this event. They include the Endocrine Society, Pesticide Action Network, and the International Pollutants Elimination Network, or IPEN. And my name is Pamela Miller, and I serve as co-chair of IPEN. The purpose of this side event is to inform POPRC members and observers about using the science of endocrinology and epidemiology to evaluate the hazards of endocrine disrupting chemicals and other related adverse effects of persistent organic pollutants. We hope that this information presented here will assist the committee members in the evaluation of substances under current review, as well as those to be evaluated in the future. This event is being recorded and will be posted on the BRS website. And now I have the privilege of introducing our distinguished speakers today. Their full bios are provided on the flyer announcement of this event. I'll introduce them in the order of their presentations. Dr. Thomas Zoeller is Professor Emeritus at the University of Massachusetts Amherst and is a visiting professor at Örebro University. His research focuses on the effects of chemicals on human health by interfering with endocrine systems. Dr. Megan Horton is an associate professor in the Department of Environmental Medicine and Public Health at the Icahn School of Medicine at Mount Sinai. She is an environmental epidemiologist with expertise studying the impacts of exposure to neurotoxicant chemicals throughout gestation and early childhood on brain development and cognitive and behavioral outcomes. Dr. Bergman has been conducting organic environmental chemistry research since 1975, focusing on human and wildlife exposures to organic contaminants, including both persistent and semi-persistent chemicals. He is affiliated as a professor with Örebro University and Professor Emeritus at Stockholm University. Thank you all. Really appreciate you presenting today and sharing your expertise. And Tom, I will turn over the floor to you. Thank you very much.
Okay, thank you. So first of all, I'd like to kind of go through what my main points are going to be, and that is to explain why endocrine disrupting chemicals are different. That is, how they have to be viewed from a regulatory perspective, view the evidence in a little bit of a different way. The first is that the toxicity is receptor mediated, and this has several implications that I'm going to go through. The second is that low doses really matter. Third is that endpoints to be employed as predictors of safety or of harm are not the usual OECD test guideline kinds of endpoints. And finally, the timing of exposure defines the endpoint that has to be employed to identify that point of departure. So let me say a little bit about the Endocrine Society. Even though it's based in Washington, D.C., it's over 100 years old with 18,000 members from 120 different countries. These are basic researchers, clinical scientists, and clinicians in practice that are focused on endocrine science. These members include scientists who really defined what we know about the endocrine system, what hormones are, what they do, and how they do it. We've also been involved in national and international policy efforts, really to focus on the science itself, to advocate for the use of endocrine science in regulatory toxicology. Now, my own focus is on thyroid hormone and brain development, and I've been working on this since really the 1980s. But I've also worked on EDC effects on thyroid hormone action and its impacts on brain development. And this is just a little bit of the reasons why I've come to some of the conclusions I've come to. The first is I was a member of the EPA's Endocrine Disruptor Screening and Testing Advisory Committee back in the 1990s. I was later on the Chartered Scientific Advisory Board for EPA and chaired the Exposure and Human Health Committee of that board. I've been on several NIH study sections as well, and the conclusion from this is that basic endocrine science really has to be applied to regulatory toxicology. Both the insights that we gain from understanding the endocrine system, but also the way we identify points of departure in risk assessment, for example. Now, it's important to recognize that EDCs have been linked to chronic disease by a number of study types, epidemiology, a variety of experimental types. This has really documented effects of environmental chemical exposures on cognitive function, on brain-based disorders like autism, attention deficit, various cancers, as well as the ability of therapeutics to address cancers, certain kinds of cancers. Obesity and metabolism are also targets of environmental chemicals, as well as both male and female reproduction, as well as puberty. And we need better regulation to protect public health in these domains. So why do EDCs require this kind of special appreciation? And the first is that hormones act on receptor proteins. And the point that I'd like to make comes from a classic view of hormone receptor interactions, where the concentration of the free hormone combines with the concentration of the free receptor to form a complex that's governed by both association and dissociation constants. We've all thought about these kind of things, but it's important to remember that the concentration of free receptor is just as important as the free hormone. And I want to show just a single graph to illustrate this. This is an experiment that was done with a number of cell lines that had different amounts of receptor expressed in that cell line. And the agonist was used to produce...
Sorry, Robert, sorry for
Yes.
disturbing you. Are you changing your slides?
Yes.
Because we can't see here from Interactive, so something's not working.
Okay.
So perhaps,
So-
yeah, now yes, we are in receptor. Yeah, that's good. Thanks.
So you can see this.
Receptor abundance is an important factor.
Okay.
Yeah. Yeah, good. Thanks.
Okay. Well, so if you don't see this graph, it's kind of hard to understand what I'm saying. So in this experiment, different cell types had different abundance of receptors. And if you look at a single dose of the agonist, you can see here that the greater the amount of, or the abundance of receptor, the greater the response. At the same time, you can read it from right to left. That is, if you take a single response, like 40% in this case, and read across, what you find is that it takes less and less of the agonist, not just a small amount, but orders of magnitude less to produce the same response. And to kind of illustrate this with an experiment, two experiments that were done, both done at the National Center for Toxicological Research. Some of the same people are on both papers. In the upper paper, they evaluated the toxicity of bisphenol A, and the low L of BPA was about 100 milligrams per kilogram per day based on uterine weight. In contrast, the second paper shows that the low L was about 2.5 micrograms per kilogram per day based on the expression of the beta estrogen receptor in the brain. That's about 40,000 times lower. Now, while you might be concerned about just the expression of a receptor, the answer was that it was caused by a different number of cells in the brain that express the beta receptors. So this is actually an adverse outcome. So the point of departure for risk assessment can lead to very different levels of protection by using appropriate endpoints. Second point that drops from this kind of analysis is that affinity is not potency. So we often see these kind of isotherms on the left where we look at the ability of ligands to displace, in this case, a radioactive hormone. And it's important to remember that this kind of affinity or this kind of analysis can't tell us about the efficacy of an EDC on different endpoints. And on the right, there's kind of an analysis based on uterine weight. And we've already seen that uterine weight can be a very insensitive measure of estrogenic disruption. So these two things are really important to keep in mind. So if affinity is not the same as potency, then the endpoint really has to matter. Different cells and tissues have different amounts of various elements, not just receptor, but different parts of the signal transduction pathways. And different endpoints will exhibit different sensitivities to the hormone or the EDC. And this potency profile across endpoints may not be the same for hormones as for an EDC. It really depends on how what the molecular initiating event is for that EDC. So also dose response shape is nonlinear always and often non-monotonic. And this is also an important point. There are many different shapes of hormone dose responses as well as chemicals that interfere with hormones. One of the most important, I think, is high dose inhibition. You can, in quotes, burn out a signal pathway so that you get desensitization rather than a continuing increase in the response to the EDC. It's so well known that, in fact, this is the basis for some pharmacotherapies for cancer, both breast cancer and prostate cancer. Signal pathways also can crosstalk in vivo, causing these kinds of different dose response shape. But an important point for me is that a non-monotonic dose response is an empirical observation, and we don't really have to have a mechanistic understanding to acknowledge that those are the data. I think oftentimes we construct ways of ignoring data rather than simply being empirical about those data. Also, endpoints for hormone action are really specific and they change throughout development. Now, this comes from my field, where this is a mouse embryo that has been made to transgenically express a blue color where the thyroid hormone receptor is being activated. In panel A and B, this is an 11-day-old embryo. Panel C is a 13-day-old embryo. If you look at the retina itself, it's quite robust expression of thyroid hormone receptor here. And we know thyroid hormone is important in retina development as well as cochlea. You can see the cochlea here. You don't see either one of those on day 11. So what this means is that the timing of exposure of a toxicant must match the kind of endpoint that you're going to look at. We wouldn't look at exposure on day 11 and expect to see an effect on the retina, for example. So to test EDC effects, the exposure timing has to be appropriate, and the endpoints have to be selected that are valid based on the specific hormone system under study. All hormone systems change throughout development. If they're interfered with, those effects can be permanent, and certainly with thyroid hormone, we know that thyroid hormone insufficiency or interference produces permanent effects on cognitive function, especially. So I want to give you some examples from the field of thyroid disruption to kind of illustrate these points. The first is the way we think about the hypothalamic pituitary thyroid axis, even clinically, is that there's a hypothalamic drive mechanism from thyrotropin releasing hormone that stimulates the pituitary to produce TSH, thyroid stimulating hormone, or thyrotropin, that then stimulates the thyroid gland, and thyroid hormones, really T4 but not T3, exerts a negative feedback on both the hypothalamus and pituitary. And the assumption here is that changes in serum thyroid hormone concentration reflects changes in tissues. But there are four important kind of additions to this model that we really have to think about. The first is that thyroid hormones are carried through the blood to different targets. And there are specific serum binding proteins that perform this function. There are also cellular transporters that move thyroid hormones across membranes. Thyroid hormone with large iodine atoms simply don't move across lipid bilayers. There are also deiodinases that convert thyroid hormone from different forms. And also this can be very local kind of metabolism. So T4 is the main hormone that's being produced by the thyroid gland and secreted into the blood. It's converted locally to T3, which is the hormonally active form. There are different enzymes that convert T4 to T3 in different sites. There's a third deiodinase that converts T3 to T2, so it deactivates that hormone action. In addition, there's two different thyroid hormone receptor classes, alpha and beta. It's interesting that negative feedback is performed by the beta receptor. So TSH, as a marker of negative feedback, is only a marker of thyroid hormone action on the beta receptor. This is important clinically, but also toxicologically. It's also important to recognize that human genetic studies have demonstrated that deficits in specific proteins that we've been talking about can produce debilitating human disorders. And in this case, it's the MCT8 protein here in the blood-brain barrier. When that's deficient, T3 is not transported either into neurons from the blood-brain barrier or from astrocytes into neurons. And we know that this shuttling is important. In the case of this genetic disorder, the consequences are severe, and it comes from just a single deficient protein that transports T3 across membranes. If there's a chemical in right now, we don't know that that's true, that produces this effect, it would produce deleterious effects, adverse effects in the human population without affecting thyroid hormone levels. So it would be invisible to this kind of screening where you're only looking at thyroid hormone levels. So this has been put together by Leo Tresande, where he looks at the hypothalamic pituitary thyroid axis within the context of the system that delivers thyroid hormone to the right place at the right time. And he has added the kinds of chemicals that can interfere with each one of these steps. And it's really important to think about chemicals that can interfere with thyroid hormone, because it doesn't really take much during pregnancy and during development for thyroid hormone action, if it's interfered with, to produce adverse effects. This comes in part from Tim Korovar and the group in Rotterdam, who has shown that thyroid hormone levels, and this is free thyroid hormone during pregnancy in the first trimester. And if you look at the IQ of the children that are generated from those pregnancies, there's a non-monotonic dose response between free thyroid hormone concentration in the first trimester and cognitive measures of cognitive function here, but also changes in the proportion of gray matter and the cortical volume is also affected by thyroid hormone levels in the mother. Now, it's important to recognize that the fetus during the first trimester can't make thyroid hormone itself. So the fetus is solely dependent on maternal thyroid hormone. From our old lab, this is kind of a first foray into this field. We know that PCBs can cause a reduction in thyroid hormone levels during development, shown here. And on the right, there's 3 graphs here, essentially. One is postnatal day 5, postnatal day 15, and postnatal day 30. We looked at the expression of a gene that's important for hippocampal function in each one of these cases, and you only see effects on postnatal day 15 and only in certain brain areas. Now, it's important to recognize that we knew when we designed this study that this gene is regulated by thyroid hormone in the hippocampus only on day 15, not on postnatal day 5, not on postnatal day 30, and only in these brain areas, suggesting that while PCBs caused a reduction in thyroid hormone levels in the blood, it was increasing the expression of a gene in the hippocampus that's important for function. Through a variety of experiments, what we showed is that 2 PCB congeners, 105 and 118, could be hydroxylated by CYP1A1 and then bind to the thyroid hormone receptor and drive gene expression. So to end, it's important to recognize that we've come up with 10 key characteristics of endocrine disruption shown here. This is in a paper by Lamerrill et al. In Nature Reviews. So EDCs can interfere with hormone action just in some way. It always winds up affecting hormone receptor interactions, but it can be caused by a number of molecular initiating events. Measures of EDC actions have to be designed based on what we know about how the system works. It's important to recognize that chlorpyrifos, chlorinated paraffins, and dechlorine plus all have EDCs activity. And I'll leave it there. Thank you.
Thank you so much, Tom. Really appreciate it. And Megan, I welcome you to share your presentation now. Thank you.
And thank you to the POP Review Committee for the opportunity to present and the invitation by IPEN, PAN, and the Endocrine Society. I will dive right in, talking specifically about chlorpyrifos, a neurodevelopmental toxicant that acts possibly through disruption of thyroid function. Next slide, please. And this is perfect. What is chlorpyrifos? Chlorpyrifos is an organophosphate insecticide originally developed as a nerve gas agent used in warfare. In 1965, the United States registered chlorpyrifos as an insecticide to replace the known toxicant DDT. This was soon considered a regrettable substitution, whereby a chemical banned substance is substituted with another chemical potentially as harmful or in some instances even worse than the original toxicant. Chlorpyrifos is a very effective insecticide used in agriculture and residences to fight both foliage and soil-borne pests in agriculture, cockroaches, mosquitoes, stink bugs, and other pests in the residence. After regulation, it quickly became one of the most widely used residential and agricultural insecticides for decades in the United States, until in 2001 and 2021, the US EPA restricted use in residential and agricultural settings, respectively. Despite its ban in the United States, it is still one of the most widely used agricultural pesticides worldwide, including in the European Union. Next slide, please. This recent publication by Hertz, Piaciotto, and colleagues leverages the most comprehensive and timely global database on recent pesticide use, including information from 71 countries in 5 different regions. So this map shows the average tons of organophosphate, including chlorpyrifos pesticides, used in agriculture by country from 2010 to 2015. The darker shading indicates a greater usage. Note that the gray areas does not indicate a lack of usage, rather a lack of information about pesticide use. In general, this graph shows, or this map demonstrates, widespread use of OPs in agriculture as well as in homes, parks, schools, hospitals, golf courses, and other public spaces that has led to widespread human exposure worldwide. Next slide, please. While occupational applications of chlorpyrifos, particularly among vulnerable populations and those without appropriate personal protective equipment, can increase exposure risk, concern for neurodevelopmental toxicity stems largely from non-intentional environmental exposures, especially among children. Babies and developing fetuses are exposed to 5 times more chlorpyrifos than what the EPA, the U.S. Environmental Protection Agency, deems safe, and children consume chlorpyrifos at 15 times the EPA standard. Exposure to children can occur through residential use, as I mentioned, largely for cockroaches, stink bugs, and for mosquitoes. Parental take-home, proximity to farmlands where there's considerable drift resulting from agricultural chlorpyrifos applications, contaminated drinking water, and contaminated foods, including fruits, vegetables, and dairy. Next slide, please. Children are not just little adults. Their physiological and behavioral characteristics render them more vulnerable to environmental insults. Evidence demonstrates that chlorpyrifos readily crosses both the placental and the blood-brain barriers, increasing exposure in vulnerable populations. In a child and a developing fetus, the detoxification and repair systems are underdeveloped. That developing fetus or child is unable to metabolize and clear chlorpyrifos, and if there is damage, they're less able to repair those systems during development. Because of rapid growth of organ systems, and particularly of the brain during early development and early childhood, this is considered a critical window of vulnerability. In general, an organ system that is undergoing development is considered more vulnerable to perturbation that may last, and perturbation which may derail the developmental trajectory of this organism, increasing the risk for adverse outcomes later in life. With exposure occurring early in life, there's a longer time for adverse effects to develop, and chlorpyrifos may have latency or persistence of these effects. In addition to differences in physiology, there are differences in behavior in young children. Young children eat more and drink more per body weight than an adult, and they eat different things, including more contaminated foods, such as fruits, juices, and dairy. And they breathe at a higher respiratory rate, meaning that if the exposure to chlorpyrifos and other pesticides is through inhalation, they're exposed to higher levels of those through their higher respiratory rate. They also have different behaviors. They have increased hand-to-mouth activity, meaning non-intentional ingestion of contaminated dust would be higher, as well as being lower to the ground, which increases their risk of inhalation and non-intentional ingestion of chlorpyrifos. Next slide, please. How does chlorpyrifos work on the developing brain? What are the mechanisms of action? Next slide, please. Chlorpyrifos toxicity is associated with irreversible acetylcholinesterase inhibition. This is the mechanism of acute or chronic systemic or acute systemic toxicity, and also the insecticidal action. So what is acetylcholinesterase? Next, please. Next. Sure. Acetylcholinesterase is a neurotransmitter at neuromuscular junctions, both in the motor system and in the central nervous system. So acetylcholinesterase, in yellow, breaks down acetylcholine to terminate this neurotransmission. So acetylcholine is acting in the synapses in nerve cells, promoting this rapid transfer of information down the nerve. Once that system is propagated, it needs an enzyme such as acetylcholinesterase to break down and terminate this action. OPs such as chlorpyrifos, next slide please. OPs such as chlorpyrifos, back one, let me finish this. OPs such as chlorpyrifos break down the activity of acetylcholinesterase, prolonging or stimulating that contraction. So there's repeated contraction resulting in death of the insect or delays in or alterations in acetylcholine function. Next slide, please. Concern about neurodevelopmental toxicity is less focused on acetylcholinesterase inhibition. According to the US EPA in 2016, there's sufficient evidence that neurodevelopmental effects occurring at chlorpyrifos levels are well below those required to cause acetylcholinesterase inhibition. So neurodevelopmental outcomes are associated with levels lower than those required for systemic toxicity or insecticidal activity. In the next slide, thank you. I identify, and several reviews have discussed potential mechanisms associating low-level chlorpyrifos exposure with adverse health outcomes. These include alterations in DNA, RNA, and protein synthesis, interference of other neurotransmitter receptor activity other than acetylcholinesterase, altered enzyme activity, perturbed signal transduction, alterations in neural differentiation, the generation of reactive oxygen species, and increased oxidative stress. And critical to the consideration in the conversation today, one of the mechanisms associating low-level chlorpyrifos exposure with neurodevelopmental outcomes is thyroid hormone disruption. Next slide, please. As Dr. Zoeller reviewed in his presentation, the thyroid is the endpoint of the HPT axis, or the hypothalamic pituitary thyroid gland axis. Next slide. The HPT axis acts primarily to maintain normal circulating levels of thyroid hormones, which are essential for biological functions in all tissues, including cardiovascular systems, bone development, liver function. It also regulates food intake and energy expenditure. Next slide. Critically, another primary function of the HPT and thyroid hormone is regulation of brain development. And altered HPT action function has been associated with learning and cognitive deficits, IQ changes, and memory deficits. Next slide, please. Thyroid hormones are essential for the development of mammalian brain activity, and thyroid hormones act on migration and differentiation of all neural cells, synaptogenesis, and apoptosis. This figure on the left demonstrates the extent and timing of these different neural processes that extend and overlap during that early gestational period and can extend all the way through adulthood. Several experimental studies have shown that low-level chlorpyrifos exposure during these critical windows of development, particularly early gestation, have been associated with changes in thyroid hormones. So chlorpyrifos exposure at dose levels not inducing acetylcholinesterase inhibition caused thyroid hormone alterations in mice dams and in their offspring. Gestational exposure in mice is also associated with significant reduction in brain thyroxine levels from juvenile stages through adulthood, suggesting that these changes are persistent. And finally, short-term low-dose oral exposure in female rats increased thyroid hormone levels in the offspring, but not in the female rats themselves, demonstrating that low-level exposures to these compounds during pregnancy can impact the offspring persistently throughout development. Next slide. So, concern for neurodevelopmental toxicity stems largely from these experimental studies and also from epidemiological studies. In the late 1990s, Gillette and colleagues overcame many of the difficulties of studying pesticide exposure by enrolling children living in two communities in the Icahn Valley in northwestern Mexico. These two communities were similar on all aspects, including sociodemographics, education of the mothers, and occupations. However, one community was highly exposed to agricultural pesticides while the other was not. As part of the assessment, children were asked to draw a person. The differences in this image demonstrate how a child raised in a non-exposed community differed from that of a child raised in an exposed community. So these are children who are average 4.5 years of age who are asked to draw a human. This study led to decades of well-designed epidemiologic studies demonstrating the adverse impacts of early life exposure on children's health. Next slide, please. Several systematic reviews and multiple epidemiologic studies following this study demonstrated that children born to mothers who were exposed to chlorpyrifos prenatally or in early childhood demonstrated abnormal prenatal reflexes in newborns. So this is a lack of the normal startle reflex in children, which is critical to development. Children demonstrated mental and motor delays in preschoolers. School-age children demonstrated decreases in working and visual memory, processing speed, verbal comprehension, perceptual reasoning, and IQ. So this is demonstrating that these effects are persistent and longitudinal across childhood. And finally, children with chlorpyrifos exposure prenatally or in early life demonstrated elevated risk for symptoms and diagnoses of attention deficit hyperactivity disorder, ADHD, as well as autism spectrum disorders, ASD. Next. Notably, these epidemiologic studies and systematic reviews looked at studies that had chlorpyrifos exposure lower than those required to prohibit or inhibit acetylcholinesterase activity. These were done among different diverse populations spanning both urban and agricultural settings. And these are done worldwide, not just in the United States or in regions known with agricultural exposures. Next slide. Perhaps one of the most profound human experiments, a natural experiment demonstrating early chlorpyrifos exposure, was done in New York State by researchers at Columbia University. In 1996, New York State passed the first pesticide reporting law, which was one of the first pieces of legislation that allowed researchers and allowed people to understand the description of type, intensity, and geographic variation of pesticide use in the state. In 1998, researchers leveraged this data to look at the total gallons of pesticide products applied by commercial applicators in the state. And so this is a map of New York State demonstrating the usage of pesticides by licensed applicators in the years preceding this study. The larger the red dot, the greater the number of gallons of pesticide applied by county. What researchers anticipated was that in the northern and western regions of New York State, which are largely agricultural, they would see greater number of gallons of pesticides applied. But what they saw is that down here in the southeastern corner of New York State, in New York City and surrounding regions, there was actually more chlorpyrifos applied than in any other county. Oh, next slide, please. The highest use of pesticides in New York State was in New York City and the surrounding metropolitan area, and more chlorpyrifos was applied in New York City than in all other New York counties combined. Chlorpyrifos was the insecticide most widely applied in New York City, and particularly by pest control operators in New York City public housing. This information was released at about the exact same time that the Columbia Children's Center was kicking off their longitudinal birth cohort study, the Mothers and Newborn Study. This is one of several NIH-funded centers focused on children's environmental health. In this study, over 725 women living in northern Manhattan were enrolled into a study to look at environmental exposures and neurodevelopment. Moms wore small backpacks during pregnancy to look at exposures in their breathing zone, and biological samples were collected during pregnancy and throughout childhood to document and quantify environmental exposures. Next slide. In this map, you're seeing this is the New York City metropolitan area with Manhattan in the middle. These blue dots represent anonymized addresses of the participants enrolled in the Mothers and Newborn Study, overlaid on a map of poverty. So you can see that these women were living in areas that were well below the federal poverty line. When queried by the study, over 90% of these moms reported using pesticides during their pregnancy. Next slide, please. As I mentioned, this is somewhat of a natural experiment. We can't ethically expose moms or children to pesticides to see what happens. However, because of US EPA regulatory actions occurring in the middle of the enrollment into the study, it ended up being somewhat of a natural experiment. So as I mentioned, enrollment into this study took place between 1998 and 2006. In 2000 and 2001, the US EPA, based on prior experimental data and increasing epidemiological data, banned the residential use of chlorpyrifos. At this time, about a third of the moms had been enrolled into the study, so this gave us an opportunity to look at the health effects of chlorpyrifos in moms that were enrolled prior to the ban compared to those enrolled following the ban. Next, please. These graphs demonstrate chlorpyrifos exposures measured in the mom's personal air samples. So again, those samples that were collected in the personal air monitoring samples and in umbilical cord blood samples collected at delivery. And what you can see is from 1999 to 2005, the dramatic increase in chlorpyrifos detected in personal air samples, with that red arrow indicating the timing of the regulations. In the graph of umbilical cord blood samples, you can see a similar decrease in chlorpyrifos detected in cord blood samples upon delivery. And in 2003 and subsequent sampling, there was no chlorpyrifos detected. So this is a demonstration of the efficacy of those regulations at reducing both environmental and internal exposure to chlorpyrifos in this population. Next slide, please. Despite reductions in exposure, we still see persistent effects of early life chlorpyrifos exposure on brain development. So this is largely looking at those children who were more highly exposed during pregnancy, so prior to the ban. What these graphs are showing is the association between chlorpyrifos measured in maternal and/or cord blood at delivery with IQ scales measured in children at 7 years of age. So in this first graphic, you're looking at the association between chlorpyrifos and full-scale IQ. So this is a composite measure of IQ in these children, and the black line indicates a significant decrement in IQ in children that were more highly exposed to chlorpyrifos. In the four panel graphics, these are looking at specific aspects of IQ, including working memory, verbal comprehension, perceptual reasoning, and processing speed. Next slide. It's important to note that these decreases in IQ are small but significant. So for each standard increase in chlorpyrifos, full-scale IQ declined by 1.4% and working memory declined by 2.8%. While these are small on an individual level, these are profound and significant on a population basis. And if you think about IQ as a bell-shaped curve, the consequences of early life environmental exposure to chlorpyrifos would shift that curve down several points, very similar to something like low-level lead or environmental tobacco smoke exposure. So it may not be impactful on an individual level, but on a population level, this is significant. What's interesting and important about this epidemiological study is that the results were repeated in several other birth cohort studies in the United States using different populations and slightly different exposure metrics. Next slide. In the Chamaco study in California, the Chamaco stands for the Center for Health Assessment of Mothers and Children in Salinas. This is the longest running longitudinal birth cohort study of pesticides and other environmental exposures in a farm worker community. So these are children living within the proximity of agricultural settings, so are exposed both through drift and through parental take-home. And again, researchers looked at measures of IQ in 7-year-olds and saw very similar effects as we saw in the Columbia newborn study. So this is again looking on the x-axis at a measure of chlorpyrifos exposure, looking at dialkyl phosphate metabolites, which is a proxy of exposure to chlorpyrifos, and effects on working memory, processing speed, perceptual reasoning, and full-scale IQ. Next slide. In a third study here in New York City, again looking at the effects of organophosphate and chlorpyrifos exposure on cognitive development, we saw a parallel or support for the same effects that they saw in the California study and at the Columbia study, primarily associations between dialkyl phosphate metabolites and adverse effects on IQ and perceptual reasoning. Next slide. One of the most critical and impactful pieces of epidemiological research that's supporting this association between early life exposure and neurodevelopmental outcomes comes from the Columbia Children's Center Mothers and Newborns MRI study. So this is an opportunity to look not just at behavioral and cognitive outcomes measured in the children, but to actually look inside their brains and see what's happening with chlorpyrifos. So researchers at Columbia University selected a subset of subjects from their mothers and newborn study and performed magnetic resonance imaging when they were between the ages of 7 and 11. Next slide. Magnetic resonance imaging allows a safe, non-invasive way to look mechanistically at the association between chlorpyrifos exposure and brain outcomes. In this pilot study that was published in 2012, researchers selected 20 subjects from the Columbia Children's Study who they considered non-exposed controls. So these children did not have chlorpyrifos detectable in their cord blood samples or in their brain samples, and in their umbilical cord blood samples, and they selected 20 age and sex matched highly exposed brains, and they looked at the difference between the exposed brains and the control brains. Next slide. What researchers saw was a rainbow of effects. What you're looking at is the right and left hemisphere of the brain, and the colors indicate either changes in gray volume or cortical thickness, or gray volume or cortical thinning. So if we're looking at changes that are in the red, we're looking at reduced cortical volume. And if we're looking at changes in blue, that's actually thickening or scarring, suggestion of scarring or alterations in normal cortical volume. So we saw significant reductions in prefrontal cortex volume in kids that were more highly exposed to chlorpyrifos. This is particularly interesting because if there is a home for IQ or working memory in the brain, it would be in these regions identified in the MRI, including the mesial prefrontal cortex and the orbital frontal cortex in the front of the brain. Next slide. Also quite suggestive of a clear association is the suggestion of a dose response between chlorpyrifos and changes in these surface measures. So among kids that were exposed to chlorpyrifos, researchers looked at the association between chlorpyrifos levels, as you see on the x-axis of this graph, and the volumetric changes in those brain effects, and saw that there was the suggestion of a dose effect or a dose response between chlorpyrifos exposure and surface measures on the brain. Next slide. So in conclusion, evidence demonstrates that low level of chlorpyrifos are damaging to the developing brain. These adverse impacts can include delayed motor, cognitive, and behavioral development, as well as structural brain abnormalities. These effects may be persistent, i.e. permanent, throughout childhood. Based on this sound science, along with 2 decades of risk assessment and input from scientific advisory panels, in 2001, the US EPA concluded that chlorpyrifos is unsafe at any detectable level and banned agricultural use, adding to existing residential bans. Thank you for your time and your consideration. That's my last slide.
Thank you so much, Megan. And Oka, I welcome you to now share your presentation.
Okay, thank you. Next slide, please. And next slide, please. Going to the science. Listen to the science. Just giving some very brief background that is basically known. Short, medium, and long chain chlorinated paraffins are what we are talking about. Maybe what is less well known that we are talking about more than 1 billion theoretical number of congenaries of chlorinated paraffins going from C10 to C30, and compare that with PCBs on 209, it's really a fraction of what we are talking about here. And we're also talking about a global production of more than 2 million metric ton for chlorinated paraffins being used as plasticizers, for flame retardants and other additives as well. Next slide, please. Well, I'm not going into so much about what the background is on persistency because they are extremely stable molecules under abiotic conditions. They actually can degrade above 200 centigrade, being a bit conservative in that. The CPAs have extremely high log KOV values, and there's much more known about the chemicals. So I'm going to the more important things that I actually want to stress. Next, please. And it's nothing new. We have studied chlorinated paraffins since the early 80s, and they are so typically lipophilic chemicals. Radiolabeled compounds that we synthesized in the lab in 1981 are showing exactly the pattern of any other persistent organic pollutants, basically, that are not hydrophilic. It's the subcutaneous fat, it's intestinal fat, and so on. Exactly what is shown for both a short chain compound here, C12 with 68% chlorine, and also for a longer medium chain, C16. It's a polychloroalkanes that was labeled with carbon 14 in Japanese quail, and you again see it in the fat, in the liver, in the lung, but you also see it in the hypophysis. Next slide, please. And these early works, we had enormous problem working on the analytical methodology, but even with the methodology we had at the time, we identified and saw, for example, moose concentrations up in the ppm levels already in the 1980s, in the, around, samples taken around Sweden. Next slide, please. Next, yeah. And it took time. It took very long time in order to improve the methodology, and actually technology saved us. Now we have methodology on the mass spec side that is reliable. And we see from this study published in 2015, the increasing concentrations of short and medium chain chlorinated paraffins in Chinese wildlife. This is a finless porpoise. But there's much more. Next, please. What is terrifying is a study that we pursued a couple of years ago, where we saw actually chlorinated paraffins totally dominating the picture of environmental contaminants in wildlife from the Yangtze River Delta area, where I was running a project. And you see only a fraction of the traditional other POPs in the peregrine falcon, the short-tailed mammusi, and this Asiatic tod. Interestingly, we also know that when you subdivide the chlorinated paraffins here, for example, for the short-tailed mammusi, we have everything from very short chlorinated paraffins to short, medium, and long chain chlorinated paraffins. So going to the next, please. But it's extremely complicated. As I said, we are talking about different chain lengths, but with the methodology we have today, it's possible to actually identify the degree of chlorine in the C9, C10, C11, C12, so on and so forth, up to 20 and 22 and more. This is human data. What we do not see, if you're looking into one of these samples, for example, from the Stockholm 216 data, within each of the chlorine degrees of the number of chlorines in the compound, we do not know the substitution pattern. So there's a large number of isomers within each one of these bars that you see as holding the chlorine degree of 5, 6, 7, and so on and so forth. So it's still extremely complicated. Next, please. The study was followed up actually by a quick shot on the concentration of chlorinated paraffins and other persistent organic pollutants in a few persons in Sweden. And what we saw from the plasma samples was very high concentrations. It's if you remember the moose data, we are above 1 ppm. It's around 1,000 to 1,400 nanogram per gram fat in the plasma. This is, when we go to the next slide, in contradiction to, if you look to the right side here, we are looking at paraffins, chlorinated paraffins in mother's milk. It's from China, it's from Stockholm, and it's from the very, very northern Norway, Bodø. And the concentrations are far lower than what I just showed in the previous picture and the previous slide. This is also confirmed by Wang and co-workers, showing fairly reasonably similar concentrations of the chlorinated paraffins in breast milk from humans in China, but also the human placenta has not extremely high concentrations. This made us actually worry about our results in the blood. Next, please. So, and just saying one more thing before going to the next slide, I just want to say that we did actually identify not only the ordinary short and medium chain, we also identify long chain chlorinated paraffins in the blood of these individuals and very short chlorinated paraffins. Next, please. What we actually did is reported right now to the Swedish EPA, which is in the same paired mother's milk and serum samples. We saw far lower concentrations in the milk than in the serum, and this is significant differences. On an average, it's 10 times more in the milk, in the serum, than in the milk. And compared to DDE and PCB 153, we see quite similar concentration between the milk and the serum for these traditional POPs. Next, please. Going to the long-range transport, others have Forkamp, and in a very recent study by Jan and co-workers, we see the air concentrations of chlorinated short and medium chain in Svalbard. We see blue mussel concentration in the very northern part of Norway, Bodø, again, where we actually have the samples of the mother's milk in one of the previous slides. And for comparison, we now have data on both very short, medium, and long chain chlorinated paraffins in blue mussels from the Arctic region compared to the Baltic Sea that is contaminated with these chemicals. And the concentrations in the blue mussels are again up in almost 1 ppm in mussels. Next, please. CPs are toxic. Yes, it's evident. And this is a graph I borrowed from Wang and co-workers that published a couple of years ago. The exposure pathways are clear, but we do have effects going through oxidative stress, metabolite disturbance, endocrine disruption, and binding to biomacromolecules, which is both for in vitro, in fish, in vivo fish studies, and in rodents, which is indicating hepatotoxicity, developmental toxicity, metabolism disruption, endocrine disruption effects. Not to repeat everything, but in addition to that, we have that MCCPs have similar effect spectrum as the short chain chemicals. They are associated with increased risk of hyperfiltration in kidney, leading to kidney impairment in an epidemiological study just published. Next, please. This, in short, goes over to endocrine disruption. The major data is actually on thyroid effects, on short and medium chain chlorinated paraffins. Neurotoxicity can arise via thyroid disruption in humans, also published in the huge review by EFSA. Estrogen and glucocorticoid are also potential targets for the short chain. Read across will most likely lead to a similar observation for MCCP. All the listed CPs here affects the thyroxine transport through their binding to transthyretin. What I just showed you, the higher concentration in the blood is most likely, and I'm saying most likely, caused by the binding to transthyretin of these chemicals, just as PFOS have shown to have done, hydroxylated PCBs, and so on and so on. So next slide, please. It's time to act. I mean, it's so evident that we have all these bases. Listen to the science, please, and act accordingly and react on all the chlorinated paraffins. I see no reason to act on short chain, medium chain, long chain individually. It's time to act on all of these. Next, please. Thank you so much. That was it.
Thank you, all the speakers. So essentially, to sum up, I think it's really important to understand and be sensitive to endocrine disruption and understand that disrupting hormone action, especially during development, will have essentially permanent effects on individuals, but also populations and societies. So I think that the information that we presented here is quite strong, and I couldn't agree more with Dr. Bergman's final remarks that we need to act on all of these. Thank you. Pam?
Yes, thank you, Tom. And I'd like to thank all of our speakers for sharing their excellent presentations and their expertise with us today. Deeply appreciated.
Could you have just one question from the floor?
Sure, if you think we have time, yes.
Yes,
please.
Thank you about the presentation. It's really useful and excellent presentations, a lot of information, but I have only one question. Each time we talk about alternative that like chloropyrifos is the alternative of DDT. And then after a while we found that it is the same toxicity and the same endocrine chemicals and make a problem. How we can deal with this problem? Each time the alternative is the same toxicity, like short chain before is safer, now it is the same toxicity like medium and long. Another question that we can ask women before marriage to make some analysis to understand if there is some exposure to endocrine chemicals or something like that. Thank you.
Yes, thank you for your question. Who would like to take this one? Very
So
good question.
I can answer at least in part. So I think the first question is, how can we avoid regrettable substitutions? And I think that there are ways to do this. There's a program or kind of a protocol called IPEN, and I can send the paper that argues this. But early on in identification of chemicals to be manufactured, there are some relatively simple and cheap ways of determining whether those chemicals have endocrine properties before it ever goes into large-scale manufacture or use. So I think that there are ways of beginning to use so-called green chemistry to avoid these kind of regrettable substitutions. The second, in terms of monitoring people's exposure levels, before marriage and pregnancy, I think is something that we really need to study. As a friend of mine once said, that statistically it's going to be very important to come to some kind of threshold or a checkbox. In addition, the genetic difference between individuals will be important. But I think we need to begin to do biomonitoring on a large scale. Thank you.
Well, if there are no further questions, I think we should conclude because I know the next session is beginning shortly. Thank you to everyone for participating. Again, thank you to our speakers, and really appreciate everyone's time today. Have a good day.